It's not live yet, Mustaq sir.
It is live. It is live.
But I'm not seeing in my YouTube.
See the left side corner, you can see the icon, red icon, and the seconds are being, the timer is on.
No, that is fine, madam. But on the YouTube where we have link, yeah, now it has come. Yeah. And yeah.
Hello. So, good morning, one and all present here. I, Mrs. Chaitali Surve, Assistant Professor, VES College of Pharmacy, welcome to you all to the 3 days webinar on product development in the era of QBD and ICH-QA, a primer for academia. Before we move ahead with the actual webinar, it is my proud privilege to introduce you to our respected principal, Dr. Supriya Shide. She is also the chairman of Board of Studies, University of Mumbai. She has over 28 years of teaching and learning experience and has many national and international publications to her credit. This activity is also an outcome of her vision and constant guidance. I request Dr. Supriya Shide, Principal VS College of Pharmacy, to address our audience. Thank you. Over to you, madam.
Thank you, Chaitali, for your kind introduction. Our today's resource person, Dr. Anil Gandhi, our coordinators, Chaitali Surve and Anita Ade, esteemed delegates, A very warm welcome to all of you to today's webinar being hosted by VES College of Pharmacy. I take the opportunity to introduce all of you to VES College of Pharmacy by taking out some time from your schedule. Vivekanand Education Society's College of Pharmacy is managed by Vivekanand Education Society, which was formed way back in 1962. Our society is well established with more than 50 years of establishment. Under VES, there are many higher and technical education which are being run. To say a few: engineering, management, law, architecture, arts, science, commerce, and polytechnic. VES College of Pharmacy was established in 2007 as a result of vision of our senior most trustee, Shri B.L. Bulani ji. We are running BPharm, MPharm program and PhD programs in the branches such as pharmaceutics, quality assurance, and pharmaceutical chemistry. And all our programs are affiliated to University of Mumbai. The vision of VES College of Pharmacy is to create professionals who can positively impact healthcare of the society and to inculcate research aptitude among our students. With this vision in front of us, we have initiated a lot of activities in collaboration with the industry. We have a collaboration center with Procter & Gamble. We have 5 MOUs with various industries. And we have a program called Campus to Corporate Transition Cell. Through this cell, every year we invite many eminent scientists from the industry and academia to interact with our students. So we are working hard to ensure that there is a holistic development of our student so that they become confident, self-reliant, compassionate, and ethically upright. So as an outcome of our efforts, we have been accredited, our BPharm program has been accredited by National Board of Accreditation, and we have a validity up to 2022 for this program. Because of our intensive interaction with the industry, in 2018, AICT and CII, in their survey of Best Industry-Linked Institute, awarded us with first prize at national level. We have been also successful in maintaining our position in the band of 51 to 75 as per the ranking framework NIRF of Government of India. Also, our VES is recognized as Scientific and Industrial Research Organization under DSIR of Government of India. So far, we have filed 6 patents and 1 patent is granted, and our faculty have successfully published 132 papers in various national and international journals. We have also received award of Best Educational Quality Enhancement Team, which was conferred to us twice by National Center for Quality Management. Coming to today's webinar, when I'm talking about the vision of VES College of Pharmacy, we have made conscious effort by appointing 2 eminent scientists as our research advisors. Professor Mangal Nagasenkar, who is a retired professor of Bombay College of Pharmacy, is now working with us as honorary professor and our research advisor. And a few months back, Dr. Anil Gandhi also had joined our team as our research advisor. He has a vast experience in the industry, and with his vast experience and exposures, he has been driving Team Westcop to do better research. So without taking much time of yours, I would now hand over the mic to Chaitali, and I would request her to take the program forward. Thank you so much.
Thank you, madam. Now a brief introduction about the webinar. A drug product's quality cannot solely rely upon its manufacturing GMP. Many quality problems are related to the way in which a product is developed. A poorly designed product may exhibit safety and or efficacy challenges, no matter how many tests or analyses have been done to verify its quality. The goal of ICH guidelines and QBD principles are to detail practices on a proactive design for product and process understanding. The speaker in a specially designed program for academia will deliberate on these principles, taking case studies of generic products and research projects usually undertaken at institute level. On day one, today that is, we are going to know more about key elements of QBD such as quality target product profile, critical quality attributes, etc. I request all the participants to write down their questions in the live comment section during the webinar so that we can take the question and answer session after the webinar. Without further delay, I request Dr. Anita Aire to introduce our speaker to the audience.
Thank you, Chaitali. Hello, good morning, everyone. This is Dr. Anita Aire. I'm associated with Vivekanand Education Society's College of Pharmacy as an associate professor in quality assurance. It is indeed an honor for me to introduce our speaker for the 3-day webinar series, Dr. Anil Kumar Gandhi. Dr. Gandhi is currently working as a consultant to many drug delivery companies in the areas of solubilization, gastro-retention, controlled release systems, dispersible tablets, ODTs, and so on. He is associated with Vivekanand Education Society's College of Pharmacy. Overall, he has more than 18 years of industrial experience in the space of novel drug delivery system and generic formulation development, analytical development, clinical studies, and intellectual property management. He is also a renowned speaker and has spoken on many forums, including ColorCon's Innovation Forum, BASF's Exact Event, Select Bio Seminar on Novel Formulation Strategies, AAPS's Discussion Forum, and so on. It's indeed a privilege for us to listen to such a distinguished person having a vast industrial experience. So over to you, Dr. Gandhi.
Thanks, Dr. Anita, for kind introduction and nice words. At the outset, I would like to thank Vivekanand College of Pharmacy and Principal Madam Supriya Madam for giving me this opportunity to organize this webinar. And dear friends, I want to just tell you that when we were thinking of having such a webinar series during this scenario when there is a lockdown and everyone is at home, and how we can take advantage of this time in terms of having some learning experience. We were looking at many topics, and then something that came to our mind was that how we can make our students ready for working in industry, or how we can make our own research work in the academia which is in line with the industry requirements, so that there is a better industry academia collaboration. And while further deliberating on these points, we decided that we should have a topic which is related to QBD and ICH, and that was the genesis for this webinar. Let's get started. This is a standard disclaimer that it's my own views only, and images are used for representation purposes. Now, when we look at the medicinal products, I want to go a little bit to the background. The medicinal products are different, and why I say that you will see from the image here. Now, as you see here, the image of fruits and vegetables, and on your right, there are some vegetables or fruits which are not so good, and on the left is those which are good. So when you go to market, obviously you're going to pick and choose the ones which are good because you can make out. Can we do this for our medicinal products? Can we do this for our drugs? Can we go to a pharmacy store, look at the blister and say, no, this is not good? No, of course not. We cannot do this. And therefore, that's why I say that medicinal products are different. Going further, I have another analogy here. And if you see, I would say many of us would have traveled in aircraft, and we would have seen that there are certain checklists that the crew members, the ground staff, everyone follows. As a passenger, I don't know whether this checklist was completely evaluated or it was partially evaluated. But I just sit and stay relaxed. Likewise, when we talk of our pharmaceutical industry, a lot of quality checks happens, a lot of process parameters are considered, A lot of systems are in place to ensure that the product that reaches a patient is a good quality product. Now, if we look at these two examples, then we will find that there is a common element, and that common element is of trust, and which is a very, very important aspect of our pharmaceutical industry. And as Chaitali madam said in the introduction, that it's not just the manufacturing GMP that will determine the quality of the product. A quality of the product is determined right from the beginning, and therefore product development is one of the key steps towards robust marketable product. And that's how I want to bring the concept and introduce you to the participants why product development is important. It is the first step in for a marketable product, for a commercial product. But if you mess, or if you not follow the right system at the beginning, then the entire product life cycle is going to be full of challenges. And therefore, it is very important that we give all due advantage to this product development step. And that's where we all come into play. We all are, important aspect of this family of formulators. Let's get started with some basic aspects. So why are we talking of ICH and QBD? So ICH basically has started a guidelines that they are able to harmonize the practices across the different geographies with respect to different aspects. If you talk of ICH Q8, which is a product development guideline, ICH Q9, which is a risk assessment guideline, ICH Q10 is the quality system guidelines, and so on. There are so many other guidelines as well. So these guidelines have an attempt to make sure that all the practices across different geographies, different companies, and at different level are followed in a harmonious manner, in a manner which is acceptable and eventually results into a good quality product. Now, going to QBD, QBD is a concept which is the scientific way of developing a product, and it works in an interdisciplinary manner across the product life cycle. So what is the emphasis in QBD? Both the product understanding and process understanding. And we will see during the course of this webinar how QBD in collaboration with ICH helps us to do a scientific product development. Now, we'll also go into little understandings of QBD. What is QBD? So QBD is nothing but a development approach, a systematic way of development with predefined objectives. So I want you to focus on the word predefined objectives, which means that I should be knowing what is my end goal, what I am developing. And this is one of the key aspects of QBD, that you have to start development with keeping end in mind. So you are very clear what you are looking at, what you are developing. And this applies again across all the aspects, whether it is a research project in academia, a research project in industry, or anything else. So a predefined objective is a very important aspect of QBD. Now, what next about QBD is that it emphasizes on product and process understanding and process control. So you will also see that there are two integral aspects of a drug product. One is the product composition or the formulation, and second is the manufacturing process. And QBD along with ICH attempts to target both aspects so that we have a good understanding of not only the product but also process. There is no point in having a very good understanding of the composition, the excipients used, the drug substance, but we don't have any control on our processes. So it has to be looked at simultaneously, like two sides of the same coin, both the product and process, and we must have very good control with respect to process. These are all necessary, important to develop a quality product. And finally, QBD is based on sound science and quality risk management. You will hear this risk word many times during this 3 days workshop or 3 days webinar, and it should also be kind of firmly fitted into your mind that at each and every step we are going to do risk assessment. And that is where the ICH Q9 comes into play. We will look at the risk assessment in much more detail tomorrow, but it is very important for you to understand that it's an important aspect of QBD. Let's go on to what is QBD and what is not QBD. So what is QBD? It's a system for managing products life cycle. It is a regulatory expectation. So if you are going to file a product or register a product with a regulatory authority, be it US FDA, be it any European authority, or many other local authorities also, they expect that you are developing product with the concepts of ICH and QBD, and you prepare your dossier also in line with the same. So it's a regulatory expectation. It is also a systematic way of carrying out research, as I've said a couple of minutes back. It also intends to increase process and product understanding. Again, we looked at it, and it is a multifunctional exercise. Now, again, this may pose a question: what do you mean by multifunctional exercise? Now, multifunctional exercise in an industry setup, you can see is that QPD involves quality assurance people, quality control people, production people, people from research and development background, and so many. So many different functionalities or different teams are associated. So QBD is not the responsibility of any one team, but it is an overall team effect where team comprises of small different departments. Now you may have a question, then how are we going to follow this in academia? Now, there is a little difference between the academic research and industry research, because in academia, we are not looking at commercially manufacturing the product, at least in the development phase. Of course, we do look at the commercialization when we partner it with industry, but at initial stage when we are doing research work, it is the understanding that a student has with respect to, say, analytical aspects, formulation aspects, and some other quality aspects is good enough with respect to the development. So this should not become an excuse that we don't have multifunctional expertise, so we cannot follow this QPD in academic setup. Now, what is QBD not there? I mean, what is not QBD? Now, very simple thing, QBD is not something new. It is more than a decade old concept. Of course, the scientific way of developing a product has been there, has been going on for ages. So only the term QBD, if you say, was coined, about 10, 15 years back, but it's an old concept. Many times, particularly in academic world, we do design of experiments, and we say we have followed the QBD. So I want to again bring to your notice that QBD is not just design of experiments. Design of experiment is one of the aspect or one of the step of QBD, and there is much more for QBD, and we will see all those things in the slide subsequently. And likewise, QBD is not just design space. QBD is associated with QTPP, CQA, and so many other aspects. So to summarize, for any researcher, be it in industry or academia, QBD must be the way of life. Now, during these presentations, you will always hear the term safety and efficacy. And these are very important aspects of product development. And therefore, I just want to make it very clear about these terms, although many of you must be familiar with this term. Now, when we talk of safety, it means that the drug product does not cause any undue harm, and it is reflected usually. What do you mean by undue harm? Undue harm is the adverse effects. So for the purpose of this presentation, a concern on safety could arise because you have higher degradation products, you have higher bioavailability, which will lead to higher plasma concentrations of the drug and leading to adverse effect. So any adverse effect, which could be a very small adverse effect like nausea, vomiting, or it could be rashes, allergic effect, or it could even result into something which is life threatening. But any such adverse effect will mean that there can be a concern of safety for the drug product. And you will see I have put safety before efficacy because safety of patient is of prime concern, prime importance. However, for certain medical conditions, you weigh the risk and benefit associated with treatment, and sometimes some kind of adverse effects are accepted considering the benefits arising from the molecule. However, those are discussed as a individual on a case to case basis. Now, if we talk of efficacy, efficacy is the capacity of the drug molecule to cause the or result into therapeutic effect or a desirable therapeutic effect. So, if you are taking an antihypertensive drug molecule, the effect that you expect is lowering of blood pressure. For the purpose of this presentation, reduction in efficacy may result due to lower assay, failure to meet dissolution, or lower bioavailability. So if you have a failure to meet dissolution, it will result into less drug into plasma or blood, and therefore it will result into not achieving desired effect. If you are looking at reduction in, say, a fever, if somebody has a fever which is very high and it does not normalize it, then you have not maybe achieved your therapeutic goal. So therefore, failure to meet any kind of therapeutic goal could mean that there is a compromise on efficacy. Now, why these two words are very important? When we are doing development, we are constantly looking at parameters which will impact either safety and efficacy, and therefore the criticality of that parameter will be determined by these two aspects. So please keep in mind about safety and efficacy with respect to a drug product when we talk about QTPP, when we talk about QBD, and when we talk about product development. So coming on to the 3 day session, we're going to do today 5 elements of QBD. Tomorrow we'll be looking at the chemical and physical nature of the active substance excipients and the way they interact with in the formulation. And finally, on the 3rd day, we will look at the process, control strategy, and design space. So these are the different elements that we are going to learn about, we are going to interact about. These are some of the references. So this is what I have referred for my presentation, these 3 ICH guidelines. And the most important one is the quality by design for ANDS, the example given by US FDA. And this is the most important document that I have referred. Most of the examples you will see, most of the tables you will see have been picked up from this example, the first immediate release dosage form example. I would strongly recommend each one of you to look at these 2 examples. They are very nicely written documents with a case study, with a product, and you can correlate to all your development activities with this. So please look at this. Most of the places I have given reference, but if there is a reference is missing, it will be most likely from one of these documents. Okay, let's begin with this background onto the first element, which is QTPP, or which is nothing but quality target product profile. Now, what do we mean by QTPP? Let's look at let's split up this term and look at the one thing at a time. So what is product profile? So product profile basically talks of product under development, or it could be a research project. What it means is, what is the profile of my product that I am going to develop? And what is the target? Target is nothing, but it talks of the product quality. So just to give you an example, that am I developing an immediate release tablet? Am I developing a capsule? Am I developing a topical gel? This kind of my target profile is what is meant by quality target product profile. So we looked at target and product profile, but what do you mean by quality? What we mean by quality is that any product profile aspect which determines safety and efficacy of the product, that will be classified as quality target product profile. Overall, we'll surely see the examples of this, but what is important here is, to give you an example, if you talk of a color of a capsule, now as long as the color used is regulatory approved color, the color of capsules will not have any impact with respect to either safety or efficacy, and therefore it cannot form as a part of QTPP, right? However, if you talk of dissolution, if you have poor dissolution, obviously it will have an impact on bioavailability, and therefore it will form a part of QTPP. If you have a route of administration, then it is very important. If you are talking of route of administration as parenteral, then the product has to be sterile. And if it has to be sterile, then, if it is not sterile, certainly it will impact the safety. And therefore, it becomes a route of administration, becomes a aspect of or an element of QTPP. Very important part of QTPP is it allows you to begin with end in mind. Let's see how it happens. But before that, let's see what forms the basis of QTPP. How do I come to know what is the QTPP or what are the elements? Of course, we will use the IR example, FDA's MR examples as the starting point. But what else we can do? First, as you all know, that most of the product development in Indian industry is the generic product development. And what do we mean by generic product development? Generic product rather is a product which is already in the market, and we are trying to develop a generic version or an equivalent version of that product. So that's the generic product development. And when we say we are developing an equivalent to a product which is in the market already, then what we are saying is that we are going to have parameters or elements which are similar to that product. And therefore, in product development, particularly in generic product development, the reference product forms the basis for QTPP. Just to give you another example or explain to you differently, if my product is, for example, paracetamol tablet, I am developing a generic version of paracetamol tablet, I cannot have a syrup as my product. It is a liquid product. So the dosage form cannot be a liquid formulation or a syrup. Dosage form has to be a solid oral dosage form and not the liquid oral. So that is the aspect that you look at it. You cannot have a paracetamol injection developed wherein you are trying to develop a generic product of paracetamol tablet. The route of administration, the dosage form changes. So that's the most important aspect, how you form your QTPP. Now you will ask me, how do we do it in the research work that we do? Because we are not doing, research work for an equivalent product. Now, typically, what serves as a guideline in this case is that you will always do a literature search. So when you do a literature search, you will know whether there are any research publications, scientific articles that serves as a basis for deciding QTPP. Also, if there is a similar product already developed in your laboratory, then that also can serve as a basis for QTPP. When do we define QTPP? Obviously, it is defined at the beginning, and you can revise based on the knowledge gained. Let's move on. I'll take a few examples with respect to what impacts and what does not. You can see a patient here. Now you will see color of tablet. Obviously, we've taken this example. Color of tablet is not important, so it will not be a QTPP. Route of administration, we already discussed. It does impact safety or efficacy, and therefore it is an element of QTPP. Flavor of syrup, again, flavor of syrup can impact the acceptability of a patient. So we have to separate or differentiate between the acceptability and safety and efficacy. You may have a bitter product, but it can be still safe and efficacious. So patient's acceptability is not so much of a priority when we are defining the QTPP. It is the safety first, followed by efficacy. Therefore, flavor of syrup is not an element of QTPP. Dissolution, again, dissolution is not very important because it is linked to the bioavailability. Same way, shape of tablet is not. Tablet assay and pharmacokinetics are part of QTPP. Let's look at one example. So you will always see, I think there's one more thing you will remember is that whenever you're doing quality by design based development, for everything that you say, every statement that you say, you have to provide a justification. And this justification aspect you will see again coming up every day in your life when you are doing following a QBD, right? So you have three parts here: QTPP element, what is your target, and what is the justification. We'll not take all of them, we'll just look at couple of them. Dosage form design, it's an immediate release tablet without score or coating. Now, why we are able to say this? Because it's a generic product and I'm following the innovator product which does not have coating and which does not have a score line. So it's an immediate release product. Now, why I'm saying that? I'm saying a justification. I can always say also as a pharmaceutical equivalent, and I will say as an immediate release design needed to meet the label claims. Route of administration, again oral. So that's the justification is pharmaceutical equivalence requirement. Stability, you will say at least 24 hours shelf life at room temperature, and this is equivalent or better than RLD or reference listed drug product, which is nothing but innovative product. There are a few other elements. You will say drug product quality attributes. Now here you will see a physical attribute appearing. We will be discussing detail in the subsequent slides about the physical attributes, which is nothing but size, shape, even flavor part coming into it. Identification, assay, content uniformity. These are the quality attributes which will impact the QTPP or the safety and efficacy, and therefore they are listed. Container closure, again, pack of the product is very important. Administration and concurrence with labeling. So you must put, that condition. How are you going to suggest the, administration of your product, which is in line with the reference product or not? If reference product says that administer with food, then even your product should mean that it has to be administered with food. Alternative methods of administration. So again, this is for a generic product. If reference product has provided any alternate method of administration, then you will evaluate it and you will also include such alternative methods in your target and provide justification. Now let's look at it. If I were to do a research project, what could be my QTPP? The elements, of course, remain same. There are about 10 elements, so we'll include all 10 elements. I want to here highlight on a couple of aspects. Dosage form strengths. Many times we are not sure what is going to be the strength. This particularly happens if I'm looking at increased bioavailability or I'm looking at a herbal product where I don't know what is going to be my dose. Now, this is something very important that I want to emphasize. When we are doing a product or a rather research project, we should have some understanding to begin with, and that some understanding must be put in the QTPP. We may change this as and when, or the more information, more development happens, more research work is done. But to begin with, we must have some information on what dosage that we are going to use. Likewise, pharmacokinetics. If I'm looking at solubilization, if I'm looking at increase in bioavailability, then I must very clearly define that I'm looking at twofold increase in bioavailability. Now, it may so happen that I do excellent work and my bioavailability goes up by threefold. That is perfectly fine, but I'm having a target, and it could work other way around as well. But I must define a target. I do see that the stability aspect many times is missing. So I would again recommend you all to put that. Container closure system also must be thought, about that. How are we going to deliver our product? Why you think is going to help? Why this QTPP is going to help? Of course, it is helping because it forces the team to brainstorm at the beginning of the project, provide clarity to students, and QTPP, you know, that can be amended. However, I want to have a word of caution here. Do not make dramatic changes to QTPP, otherwise, it makes the earlier exercise futile. So be careful. Try to make QTPP a kind of a final QTPP at the beginning of your research project. Let's go to the second attribute, which is nothing but quality attribute, which is a critical quality attribute. To get into this attribute, what is the attribute? Attribute is a product parameter or test which are specific to product under development. What it means, it is the test or product parameter. And I want you to focus here on the word product, because when we talk of critical material attribute subsequently, you will know that it is for a intermediate or starting materials. It is not for the product. Critical quality attribute is something which is for the product. And for ease of remembrance, you can keep in mind that a critical quality attribute always results into a specification or test which are used for release of drug products. So you know that it is for a drug product. What do you mean by quality? It determines the quality of the product. And why it is critical? It is critical because an attribute can impact safety and efficacy. Again, this is an important aspect of development. When we identify when we identify a quality attribute, you will see that always the safety and efficacy is kept into consideration to determine whether an attribute is critical or not. We will, of course, see in subsequent slides. Now, what can be a quality attribute? A quality attribute can be a physical attribute, chemical attribute, biological, and microbiological property. When you say physical, it means it could be like shape, size, flavor, etc. Chemical means assay, degradation products, dissolution falls under chemical. Biological means the pharmacokinetics aspects, and microbiological means the microbiological limit test. Now, what it further says that these attributes should be within appropriate limit or range or distribution to ensure desired product quality. We'll see this in little more detail in the subsequent slide. Now, you will see that this quality attribute is determined in a systematic and scientific manner, like with all the all the elements of QBD. What do we do? First is you list physical, chemical, biological attribute. Then you will put a subset. A second step is to put a subset. What do you mean by subset? In front of physical, you will write appearance, size, shape. In front of chemical, you will write assay, dissolution, and so on. What is the target? Target is what is the limit in which this quality attribute should be there. Like if you say dissolution, then you will say, 85%, more than 85% in 45 minutes is my limit. Or if you say assay, your target is 90 to 110% of the label claim. Now comes the most important aspect. Is this a CQA? What does it mean? Is this quality attribute a critical quality attribute? And this aspect is decided based on the safety and efficacy part. Now, why do we decide whether an attribute is quality attribute or not? The reason is that we are going to use this quality attribute aspect throughout the development. Now, I will not be able to do an identification test on each and every batch that I make, but I will do dissolution on each and every batch that I make, because dissolution is a critical quality attribute. So, an attribute that can impact the product development will be considered as critical because it will eventually impact the safety and efficacy. Therefore, we have to determine whichever attribute is critical, we have to determine it very appropriately and in a scientific manner. And last, as I was saying, with all QBD elements, you have to provide justification. Let's look at one more slide. After that, we'll go to the example. So, what is how you are seeing now the links. QTPP will lead to CQA, and CQA will lead to the product specifications. Product specifications is nothing but the test which we use for release of a drug product. So now you can always see the connections, that if you have a failure in the specifications, you will have the failure in the critical attribute, which will mean that it can impact safety and efficacy. And therefore, these terms are very important from the development point of view. So I think we've discussed about this aspect. I just want to highlight that it is monitored throughout the drug product development, and the CQA list can be dynamic and may be updated based on the product and process knowledge. Again, we will see why and how. This is a standard table. As you can see, I have taken it from the example of quality attribute of a drug product. What is our target? Is this a CQA? And what is the justification? I just want to take one example here so that we can complete all the slides today. So let's take the example of physical attribute, which is the friability. What is my target? Not more than 1%. Is this a CQA? No. Now, why this is not CQA? Suppose a tablet which is received by a patient is chipped. Can it impact the safety and efficacy? No, not really. Of course, it can affect the assay, but it cannot affect the safety and efficacy to that extent. And therefore, friability is not considered as critical attribute. But do I study friability in development? Yes, we do study, because you want to ensure that patient receives an intact tablet and patient does not, have any complaints associated with the product. So you have to look through each aspect in much more detail, and that happens when you have to give a justification. Let's look at one more aspect, which is a content uniformity. Now, content uniformity, if you look at the quality attribute, what is my target? Conforms to USP limit, or we have a RSD limit of, let's say, less than 5% in the assay value of each individual tablets. Is this a CQA? Of course, yes, it is a CQA. Why? Because variability in CU will affect safety and efficacy. How? If you have variability in content, there will be a tablet which can have assay of less than 90%, or there can be a tablet which can have assay more than 100% or more than 110%, which is my limit. So in both the cases, it can either affect the safety or efficacy, and therefore it is a critical quality attribute. Likewise, you can see degradation products. You have given the limits. All the terms like ACE 1, 2, 3, 4, 5 are taken directly from the examples. So if you have a degradation product going beyond this limit, then obviously it can impact the safety. So you are deciding a quality attribute first, which is listing all the attributes of your dosage form. You are giving a target, and then you are deciding through justification whether it's a CQA, that means it's a critical quality attribute, or not. I want to take here an example of a research project and look at, both the critical quality attribute and QTPP. Now, suppose a research project is topical formulation of solid lipid nanoparticle. Now, as I see here, it is not just one product or one project, but it can be split into two products, or two projects rather. First stage is the preparation of solid lipid nanoparticle, and second steps is the incorporation in the topical gel formulation. Now, I can have two QTPPs. However, what is very important is that I should have a final QTPP, which is the overall QTPP, and which is the QTPP of the final formulation. I can have an intermediate QTPP, which is QTPP for stage 1. The only purpose of this is that I am focused with my stage 1 development as well. This helps me to have a better understanding, because both are very different. Now, this does not happen typically in a tablet formulation or a capsule formulation, because even at granule stage, you are talking of the assay and dissolution. Whereas in case of solid lipid nanoparticles, just an example I'm taking, it could be a very different case. So the dosage form is different. Overall will be a gel formulation, but at my stage 1, it could be SLN particles suspended in a liquid, and so on. There can be a difference here. And what is important is the CQA. And I want you to highlight more on or focus more on CQAs, because the CQA is the one which is going to be a gate which will allow you to go from stage 1 to stage 2. So let's say you have a viscosity limit or particle size limit or polydispersibility index, which is typically, or even zeta potential, which is typically used for solid lipid nanoparticles. So this I will use as a is a gate or as a release specification before I move on to the topical product. Now, I have written some of the aspects as not applicable, but you may still include particle size of the topical gel product as well. So these are some of the examples of CQAs for a topical SLN product. You may add, or you may have a different values to it. Just take this as a case study or as an example, and I'm sure with all the people who are very well experienced in these areas will be able to customize this table. It is not it's a comprehensive table. And also I want to say that I'm not an expert in SLN formulation, so I've just put it as a table, but surely it can act as a guideline for you to think through and develop your own CQA table. Finally, I also want to emphasize that it is always possible to have such QTPP or CQA for any chemistry or herbal based products as well. Let's go to the next attribute, which is critical material attribute. While discussing CQA, I have emphasized that CQA is for the finished product, whereas critical material attribute is with respect to a specific material. So it's an attribute which means that parameter or tests that are specific to a particular material. It is related to input material or intermediate material, for example, drug substance, excipients, granules, blends, etc. And finally, criticality, it will be determined by its impact on critical quality attributes. And now you should be able to see the link. We are deciding critical quality attributes based on its impact on QTPP, and we are deciding critical material attributes based on its impact on CQA. I want you to look at these terminologies right now in isolation, kind of, because we're not looking at connecting it or with respect to the product development. I will be taking this up in tomorrow's session. The today's objective is to introduce you to these terminologies, that is CQA, QTPPs, the CMAs, and CPPs, and so on. So just to give you example, bulk density of microcrystalline cellulose, which is used as a diluent, will be a material property or a CMA. Bulk density of granules which are ready for compression will be a critical material attribute. But assay of a tablet will become a critical quality attribute because it's for the finished product. So any intermediate products or any material aspect is the critical material attribute. We'll have to differentiate and understand this very clearly. Let's look at what do we mean by material and what do we mean by an attribute. So like CQA, this is also a physical, chemical, biological or microbiological property or characteristics which should be within appropriate limit so that the quality of that material is ensured. Now, there are a lot of material that we can think of, but for the purpose of our development, three materials are very important: drug substance, excipient, and in process materials. I want you to be very clear about in process material definition, which is an intermediate step definition. Now, if I were to relook at my example of solid lipid nanoparticle, the solid lipid nanoparticle of stage 1 could actually be an in-process material because my finished product is the topical gel product, right? So anything that is not the finished my product, which is marketable or a commercial product, if you refer to any other material, that will be the material, and its attribute will be, we have to consider as a critical material attribute. Okay. Let's also look at the difference between CQA and material CMA. As we say, CQA is for output material. What do you mean by output? Output is something which we produce, which we commercialize. CMA is with respect to an input material. What is input material? Input material is any material that is consumed in the process to make output material. Okay, I hope it's very clear. CQAs are specification derived from CQA. I mean, specifications rather are derived from CQA and which are used to release the drug product, whereas CMAs are the specifications of input materials which are used for testing at the beginning of the process or starting a batch, or it is an intermediate testing before you proceed to next step. I hope the difference of CQA and CMA is clear to all. Now, what is the definition of critical here? Now, a material attribute, it could be a physical property, chemical property of any material. When we make any realistic change in that, it can have a significant impact on the quality of output material. Now let's give us some examples. Now particle size of API, which is a BCS class 2, that's biopharmaceutics classification 2, which is low solubility and high permeability. A molecule belonging to this class surely will have a dissolution as an important aspect, and the particle size will determine the dissolution. So particle size of API will become a critical material attribute, whereas the dissolution is the critical quality attribute. Now, when I look at other aspect of it, if I have a BCS class 1 molecule which has very high solubility, then particle size may not impact dissolution, and therefore it will not be a critical material attribute. I want to again emphasize here, the particle size of API is surely an material attribute. It is an attribute, but whether it is critical or not will depend on how it impacts the critical quality attribute. For example, dissolution in this case. Let's go to the next element, which is the critical process parameter. Now, the word itself, critical process parameters, refer to any parameter which is used to control a process or process step. For example, a temperature in fluid bed drying, or for example, the blending time or blending RPM or compression machine speed, and so on. This is a process parameter. Now, it of course refers to the process part of it, and therefore it's called as a process parameter. And like critical material attribute, this parameter, if it affects quality attributes, critical quality attributes like assay, dissolution, degradation product, then it will be considered as critical. To give you an example, if I have a drug which is sensitive to temperature, which means that at higher temperature it could get degraded, then the temperature of drying process becomes a critical aspect. I may set the temperature as 45 degrees and put a range as 40 to 50. Of course, I have to prove that in this range it remains stable. But if I cross this range, let's say a drying happens at 60 degrees, then I'm going to see, or I'm likely to see, increased degradation products, increased impurities, of course, which will potentially can impact the safety. And therefore, this parameter becomes critical. So any process parameter that can impact your CQA, which is assay, dissolution, and degradation product, and so on, will be considered as critical, right? And therefore, you need to control this parameter. Of course, we have looked at the examples. How do we reflect this? We'll be dwelling on this more detail on day 3, but just to give you an example, that typically in a process diagram, uh, process step which is with the dark or the bold border is considered as critical. Like in this process, if you see blending, dry granulation are the critical steps. Also compression is critical, whereas milling, lubrication and packaging is not critical, because let's say packaging is unlikely to impact the safety and efficacy, and therefore it's not a critical step. What is the correlation? Now we have very few minutes left actually. So I would like to, complete this 2 elements. This is the second last element, and we should be able to finish in time. What happens is when we are making a product, we are having the input materials like drug, excipient. This becomes input material. The attributes of this become a critical material attributes. Then we have a unit operation, let's say blending, compression, and so on. The process parameters which are important are the critical parameters with respect to operation. What it results into is the final product, and when I test it, what I'm testing it against is the critical quality attributes. So this slide gives you an overview of connectivity between CMAs, CPPs, and CQAs. Now, why we refer to CPP1, CMA1, CMA2? Because you may have multiple materials. You may have excipient like microcrystalline cellulose, you may have cross carbamylose, you may have in-process materials like granules. So the material attributes with respect to each of these materials are different, and that's why you will say CMA1, CMA2, CMA3. Likewise, you have critical processes like blending, you have critical processes like compression. So you will have CPP1, CPP2, and so on. And even in blending, you may have many parameters like blender RPM, blender occupancy, the time of blending, and so on. So this is what in nutshell is about these 3 elements of quality of QTPP or QPD. We will be dealing again with this in a lot more detail on day 3, but for you it is important at this point in time to understand what is the link. Let's come to the last part, which is the control strategy. Now, control strategy is nothing but the parameters or what is the limit that we use. Now, in 2 examples that I gave you, that if you have a particle size of drug as the critical parameter because it's a BCS class 3 or sorry, BCS class 2 molecule, then you will have a limit on the particle size like given in the table here below, D90 of 10 to 30 micron. This limit given is my control strategy. Control strategy is how am I going to control a particular material attribute? How am I going to control a process? Like I have given you example of fluid bed drying, where you set the temperature as 45 degrees, but you have a range from 40 to 50. 40 to 50 is my control strategy. I am allowing the process to run between this temperature range, but above this or below this temperature range, I know that it can have impact. So control strategy is nothing but how I control a particular material attribute or a particular process parameter. So with this, I want to come to, I'm coming to this, penultimate slide of the presentation. This is how, there is a link. You have a product quality or quality target product profile, which leads to critical quality attributes. We'll do a risk assessment, which we'll do tomorrow, resulting into the design space. The risk assessment is done on the critical material attribute and critical process parameters to arrive at design space, control strategy, and then product life cycle through continuous improvement. So this is where we are. And to end my presentation, I want to highlight that for any product development efforts, QTPP is the guiding star. QTPP is the goalpost that you want to reach to, and CQA is nothing but roadmap to achieve it. So that's my last slide. Thank you so much for your attention, and thank you so much for joining in. Stay safe, stay healthy, and yes, I'm ready to take up any questions that you might have. I want you to just note that because of the time constraint, we will take few questions, and I will surely try and get back to all of you with the questions that you may have posted in your comments. I'll come back to you on those questions. Thank you so much once again.
Thank you, sir. Thank you so much. So I think we have some time left for question and answer session. So I would like to take few questions. So first question is a very basic question. So is it absolutely compulsory or mandatory to use QBD in pharma industry?
Okay. I am glad that these questions are asked, though it can be basic, but it is very important for everyone to know this. If we talk of pharma industry, yes, it is absolutely necessary. Particularly if you talk about regulated market, if you have not followed QBD principle, your dossier may get rejected at the beginning itself. So QTPP, CQA, all elements must be there in your product development report. Now, this is one regulatory aspect of it. But what I also feel is that from scientific principles of development and from logical perspective as well, we must have these elements. It may so happen that you might be developing a product for a market where this is not requirement. However, even if it is not regulatory requirement, you should follow this, like you will follow this in your research project in institutes where it's not a requirement. So for pharma industry, I would say it is a must. For research projects in institute, I think it should be becoming must.
Okay. Okay. Thank you, sir. Next question that we have is QTPP applicable to drug substances, basically API manufacturing? And if yes, what could be the elements of that QTPP?
Again, very good question. QTPP is not directly applicable to drug substance manufacturing, but you can still design your own QTPP. Now, the QTPP is basically an element which is for a commercial product or a marketable product. What is your target profile for your market product? And if you look at the formulation industry, then the drug substance becomes a starting point. So with respect to drug substance, you will be looking at critical material attribute and not the QTPP aspect. However, the drug substance is an end product with respect to a chemical industry or a drug substance industry. So I can devise my own set of specification with respect to a drug substance. So it is, yes, it is possible. And one thing we must remember that it has to do with keeping end in mind. So what is my end goal with respect to drug substance, synthesis or manufacturing? And keeping that end in mind, I can derive my own QTPP. But as I said, for drug product industry, it is the starting material.
Okay. Thank you, sir. So the next question is there any link between QTPP and biowaivers?
Okay, very innovative questions, I feel. There is no direct link between the BioWaiver and QTPP. What I would say is that, BioWaiver is not something that you will probably document at the beginning. Yes, you may start your development considering biowaiver. So the pharmacokinetics aspect of QTPP, you will write there as the pharmacokinetically equivalent or bioequivalent to the marketed product. However, the route that you take for that to prove bioequivalence could be through biowaiver. Now, there are certain guidelines with respect to biowaivers that we must follow with respect to determining the BCS classifications, with respect to permeability, and so on. So, this will form part of your development activity, and you will justify in your development whether it is possible to go for biowaiver or not. But as far as QTPP is concerned, I will still put there as the bioequivalent to the marketed product and justify it through my research and development work in PDR later for biowaiver.
Okay. Next question is, can you just briefly again explain difference between QTPP and CQAs?
Okay. So QTPP is something which is my end goal, my target, which I am going to achieve. CQA is what how I achieve the QTPP, and CQA is one of the aspect of QTPP. This is something which is very important for us to understand that QTPP has many elements. One of them is CQA. Now, further differentiation is that CQA is what I use as my specification to release the drug product. I do not use QTPP to release my drug product. CQA will say whether a product is immediate release product or not. QTPP will say whether it is packed in aluminum blisters or it is packed in HDPE bottles. CQA does not say this. CQA will test the product and say whether it is appropriate to market or not. It is the testing that controls, or that is the key aspect of CQA. CQA I will be using throughout my development, but QTPP I will set once and then I will see to it that I meet it. When I say this, I have a CQA which is, as I was saying, dissolution. Now I'm developing a control release product, and it's a 12-hour release control release product. So I will always, all my batches that I use or that I develop throughout my work, I will be doing this, development dissolution studies on this to determine whether I have met my target. But essentially, all those products are going to be a tablet or a capsule which I have defined in QTPP. So I might be changing the component or the composition to achieve a particular CQA, but my QTPP does not change, which means that if I have decided that it's going to be a capsule, it will remain capsule throughout my development. If I have decided my route of administration is oral, it will remain constant throughout my development. So this is the QTPP. CQA is the testing, how I develop this product. I hope this is clear, but
Okay.
I can take it separately if anybody wants further clarification.
Okay, sir. So we have a question from Madhura. She is asking, how can we apply QBD principles to develop formulations which do not have a marketed product to be used as a reference?
Right. So this is something which I also mentioned earlier. When you have such a case, you should use first is scientific principle. Now you are developing something completely novel, and there is nothing such in the general practice. In such cases, obviously you will use your scientific and logical understanding, but there are other options available. Now, if you are developing, as I've taken, let's say you are developing a nano emulsion. Now, is there any research paper wherein nano emulsion technology has been used? I will use that research paper or a series of research paper for arriving at my QTPP. So my QTPP will be derived from the public domain knowledge. I've given you one more example is that if as a student you are developing such a product, and in your research lab, there is somebody, your senior has done similar work. That work, maybe at that time the QTPP was not defined, but that work can act as a guide for you to develop QTPP. So my QTPP is not something just purely based on what is there in my market. My QTPP is what I want to develop in the end. So my end goal has to be very clear. And if there is nothing available, I'm doing excellent innovative work, then I should still think, using this all available information, I should still think and draft my own QTPP.
Okay. So we have another question from Charul. So she is asking, can QBD principles be applied to developing analytical methodologies? And if yes, then how?
Absolutely. I think there is a particular, session that can be, in fact, made possible if you want to learn more about how QPD can be applied to analytical. But yes, it is certainly possible. I can have a goal in mind when I'm talking of analytical methods, and these are very simple and very effective goals. I can say that when I am developing a related substance method, my runtime should not be more than 45 minutes. That is my target. What is my target? Resolution between the impurities or between two peaks should be more than 2. There is some number on theoretical plates. So I can define my own targets before I start a method development. Then you will see tomorrow when we do a risk assessment on drug substance attributes, material attributes, the process. You can do a similar risk assessment on your analytical methods. For example, if I choose a C8 column, what is likely to be the impact against a C18 column? Or because my substance is so polar that I want to use some other column, what is going to be my mobile phase? So I know what are my parameters or attributes, and I will use my chemistry knowledge to understand how these attributes will get impacted. Now the attributes of course changes. My attribute could be my retention time of main peak. My attribute could be my resolution. So I can select all these attributes as my critical analytical method attributes, and then these attributes could be targeted for my method development. And I should also emphasize here that many companies, many pharmaceutical companies are already following this QBD principles for analytical method development. It also, let me add, it's not a regulatory requirement as of now, but we might have it coming up in future, because it's again just a systematic and scientific way of developing analytical methods.
Okay. Thank you, sir. So we have covered most of the questions. So thank you, sir, for a very informative session on day one. We are looking forward to hear you on day two and day three. So a few instructions for the participants now. The link for the e-assessment is active in the description part of the YouTube video. Please refresh your YouTube page so that you can get the link for the e-assessment. Second thing is, many people are asking for the PPTs. So this YouTube video is already available on our channel, on the channel VS College of Pharmacy. So you can anytime go and view this video again. Also, please subscribe to our YouTube channel, like it, and click on the bell icon so that if we have any upcoming interesting webinars, you will be getting notified with the help of this. So please do like and subscribe our YouTube channel. Okay. So thank you so much for this, and hope we will see you tomorrow again at the same time, that is 12 o'clock. And thank you so much for attending this interesting session.